you for that kind introduction. At the outset, let me thank Professor Jagannath Rao, CEPIPRA and KL University for the invitation to be here. I have put up this title quite general, that is Alzheimer disease and dementia. Before I start my presentation, let me also mention that I do have a strong relationship with France. In fact, I have many friends and collaborators in France across Paris, Bordeaux, Strasbourg, and also I'm part of CEPIPRA for some time. So that is the link I have with France. Thank you. So that is our broad interest. We work on the theme of functional amyloids and disease amyloid. Both the topics have relevance to dementia, but for today, probably I'll move on with this. We talked about dementia. From yesterday, the overrun, again, the dementia, the term was mentioned, but let me tell you, particularly this is for the student audience in the back. I think it would be nice if you can all move, friend, those who are sitting in the back side. Dementia is not a specific disease. I would like to emphasize, dementia is not a specific disease, rather an umbrella term used or referred to very many diseases, actually more than 25, 30 different diseases, which are characterized by loss of memory and other thinking abilities that severely interfere with one's life, particularly the patient's life. And if you look at very many diseases I talked about, the Alzheimer disease is a major contributor to dementia. In fact, it contributes anywhere from 60 to 80% of all the cases. And in addition to dementia, there is another scenario that we call it as mixed dementia. In fact, it was passingly mentioned by, I think, Senthil or somebody else as well, meaning where more than one neurodegenerative or a disease coexists. For example, for instance, Alzheimer disease coexists with Parkinson disease. We call them Parkinson plus. It makes the situation even more worse. Coming to molecular mechanisms, we are going back to 12th standard textbook. We all know that the proteins are synthesized as polypeptides, nascent polypeptides, and which are subjected to certain modifications and also what we call folding mechanism, so that they acquire three-dimensional structure and become functional. Well, these functional proteins and also their processed products, meaning I am referring to peptides, undergo bisfolding and escape repair mechanism as well as degradation mechanism and become amyloidogenic. What do you mean by amyloidogenic? Meaning they undergo ordered aggregation to produce toxic materials or protein aggregates or toxic materials in the brain and responsible for, as I mentioned, very many neurodegenerative diseases. In fact, Alzheimer is one such disease, a major neurodegenerative diseases, and of course this is named after Alois Alzheimer, who characterized for the first time. And these are the two protein plaques. I'm talking about protein aggregates. One is made up of tau, another one is A beta. I'll come to that in a while. Well, as I mentioned, in spite of there is a good progress in the area in terms of understanding as well as developing tools for managing. Still, there are no effective detection and cure for this disease, which impact public health and economy. Now, again, for the benefit of the students, when it comes to symptoms, the Alzheimer disease is mainly characterized by cognitive deterioration, which may be related to memory loss or memory, language, movements, recognition, decision making, and planning. Let me take a case study and demonstrate to you how severe Alzheimer disease can be. What you see is an artist, an American artist who lived in UK. That is his portrait in his 30s. Subsequently, in the year 1995, he was diagnosed with Alzheimer disease and he was courageous. He wanted to know how it feels like living with Alzheimer disease, and also possibly he wanted to convey to the world how it feels like living with Alzheimer disease. So we went on to do the self-portrait in the subsequent years, and those are his self-portrait. as you would see in next couple of years, how the health deteriorates. At the end, all you can, kind of scrib is a kind of a skull. So that is the severity of Alzheimer disease. And of course, I started working in this area roughly 15 years ago, and my peers, peers and friends, and they used to ask me, "What are you working on?" Well, when I said I'm working on Alzheimer disease, of course, it's not their fault, but indeed, they laughed at me, saying that Alzheimer is a disease of Western countries. So why are you working on Alzheimer disease? But let me tell you, that question is no longer asked now. It's Alzheimer disease is quite very well known in the community. In fact, there are movies in in every language. We have very many languages in India. That means public is very well aware of this particular disease. Of course, before that, there is nothing wrong with the peers because mental health was not appreciated or taken care so much in India. Of course, that is changing. That is also because of the reason that we in India we used to equate dementia with aging. But let me tell you, although aging plays a role here, but there is a clear difference between aging and dementia or Alzheimer disease. Alzheimer disease is a true disease because if you look at the memory deficit here, unfortunately this, I can see that. You see that the green trace, that is the memory in case of aging, it never falls to the baseline or zero, for instance baseline. But when somebody is detected with Alzheimer disease or a dementia, and from that point onward, there is a free fall of memory. I think it clearly tells you that aging, dementia is not, is aging, rather a true disease. Of course, coming to our own lab, so that is our programs. So we work on understanding disease mechanism, diagnostic, therapeutics, and even theranostic. We have very good success so far. Probably I'll going to give you a glimpse of some of these success stories. And of course, in terms of mechanism, we were working on role of metals, inflammasome, glia, mitochondria, and gut microbiome, peroptosis, gyprotis, and identifying non-coding RNAs and many more. And in fact, one or the other topic overlap with the people who talked about either yesterday or today, except that I don't directly use AI and ML, but these tools definitely useful for us as well. Well, now the question is, particularly in the back rows, now you might be asking, well, Alzheimer disease is another disease. Why this particular disease eluded researchers or clinicians from developing a drug candidate for this? That is mainly because Alzheimer is not a simple disease, rather a multifactorial disease characterized by multifaceted toxicity. Meaning I also talked about protein aggregates, then there are metals, copper, iron, zinc, and which actually some of these redox metal undergo oxidative redox cycle, generating lots of reactive oxygen species, causing oxidative stress, and there is a neuroinflammation, all of which damage mitochondria and biomolecules and many more such disease pathways or routes here. That is exactly makes it complex, complex, and also that's how it eluded researchers from finding that particular the magic bullet. Of course, I am not going to the details. If you are interested, particularly in the back rows, this is the book, a comprehensive book which covers many aspects of Alzheimer disease. Possibly you may like to refer. Once again, I would like to re-emphasize the fact that Alzheimer is a true disease and not an inevitable part of aging. And in the year 2018, National Institute of Aging and Alzheimer Association came up with a research framework which designated these two proteins which I talked about, which I mentioned earlier, A beta, tau, and associated neurodegeneration as designated biomarkers. In fact, this National Institute of Aging and Alzheimer Association recommend and suggest that we must diagnose Alzheimer disease based on specific biomarkers, not based on behavioral symptoms or visible symptoms. And of course, the framework is also left open-ended so that whenever we identify a new biomarker can be added into the list. In fact, we have one such addition to this. So nevertheless, some years ago, no, particularly probably there may be many biotech students, engineering students here. We all have tools, right? Everybody uses tools. In our lab, tools are molecules. So molecules are the toolbox. So for example, here we developed a near infrared molecular probe which not only detect Alzheimer disease, not only can detect Alzheimer disease, it can differentiate Alzheimer disease from other neurodegenerative diseases. It's a very, very unique tool, meaning it not only detects Alzheimer disease in its a normal condition, but can detect Alzheimer disease differentially even in case of mixed dementia. In fact, we are now taking it this forward for making it a clinical platform through our startup company. And of course, I talked about reactive oxygen species. Well, for the engineering student, I can only talk about electrons. Possibly, I can use the word electrons. Oxygen reduces to water through 4-electron system. In the process, it generates very reactive species, and these metals also plays a very, very important role. We call them penton type reactions, and of course, there is HOCl as well. Many of these reactive oxygen species, they are very, very important physiologically. However, if they cross a threshold concentration, they become, extremely life-threatening. In fact, they are very friend, but if they cross threshold concentration, nobody can be, kind of a dreaded villain than these tiny species. In fact, we developed a number of molecular tools, and we could image these multifaceted toxicity in the AD brain, of course, in the mice model. This is what you see is not just an image. What we are imaging here is actually a multifactorial Alzheimer disease or a complex Alzheimer disease. Now, for the student's sake, how do you see that? I talked about the proteins being toxic to the cells. How can we see that? Well, we can look at the cells and see how happy or how sad they are. Very simple. That is called cell viability. These cells, what you call, what you see here, you have to trust me, they are healthy cells. And the moment they come in contact with these toxic proteins, you see that's the fate of it. Of course, I will not go into the technical aspects of it. That's how the cells looks like under these amyloid aggregates, and subsequently that leads to neurodegeneration, neuronal cell, and that actually disrupt neuronal circuit or network. In fact, yesterday we talked about neuronal circuit, wiring, and so on and so forth. And precisely that's what happened. This neurodegeneration or a neuronal cell death leading to disruption in neuronal these circuits, wiring, or a network that leads to problem in cognition or learning memory disabilities. Of course, so this you don't have to worry about all of it. This is a in vitro data. In fact, you can test tube. We can see you can make these protein aggregates. And those protein aggregates indeed becomes toxic to the cells. They interact with the cell and make hole, and then leading to different kinds of toxicity. But one can also develop molecules which can prevent this kind of protein aggregation, and you can protect cells. And of course, not only look into biological or a physiological cell viability, but you can even measure their mechanical properties. This is another very interesting area. Just by looking at these mechanical properties, one can tell these cells are healthy or under disease conditions. Well, now I'm moving on. This is the status of in terms of treatment. Okay, so one earlier we used to have symptomatic therapeutics which are of no use for and then okay, yeah. And there is disease modifying therapies. Of course, we don't have much except that last couple of years. So we have 2 drugs were introduced, that is antibody immunotherapies. Of course, hugely controversial, probably the most controversial drugs ever introduced in the market. But possibly because to trigger to make the community positive and moving forward for a new discovery is probably FDA approved, and then of course it still do not give the same level of the efficacy one would expect. Of course, at this stage we realize that we need to develop multifunctional molecule, not simple molecule, multifunctional molecule which act on many of the pathways which I mentioned earlier. Possibly that is the way to tackle Alzheimer disease. In fact, this is the one approach. This is a tripeptide which is from our own body. All of us have this tripeptide. We worked on this particular molecule, we modified, and indeed we make it a multifunctional molecule. Again, I will not go into the into the detail because paucity of time, but all I would like to say that indeed, at least in the cell line, cellular level, it become multifunctional, modulator. Another approach we introduce, we talk about drug repurposing. It's become a very buzzword, fashionable word in recent times, drug repurposing. But let me tell you, here we introduce probably the next level. It's not exactly drug repurposing, possibly just to fine tune these the failed drugs or a natural product so that they become very, very effective and specific to Alzheimer's disease. What we do is we integrate structural and functional components of a failed drug or a natural product, where the failed drug is failed because of a toxicity, and then there is a natural product. Most of the time this natural product, particularly for example polyphenolic compounds, they are more nutraceutical than a drug. Okay, so for example, this drug compound, the EGCG, this is from green tea, and then people say, this has also been considered by clinical trial, but let me tell you, this can never become a drug. For simple reason, it is a nutraceutical, but if you drink a green tea for a 10 years or a lifetime, definitely there will be a good effect. But drug, to consider a molecule to be a drug, it has to work in a day, 2 or a week time, month time, or a 1 year or 2 year. You cannot wait for lifelong. So this can never be a drug. So what we did was, on the top one, what you see is a cleoquinol, one of the earliest molecule to enter clinical trial and failed because of the toxicity. What we did, we integrated the structural and functional components of these two. This is a general methodology. I'm giving you one example, and we come up with kind of a hybrid molecules, and indeed, where we reduce the toxicity and improve the efficacy of the natural product, and indeed it become a multifunctional molecule. And of course, here another example, just like a curcumin, right? You test curcumin for any disease, it works. Okay, you have to be extremely careful. There is a paper in JMC, it interferes with the assay itself. I'm not saying, see, I'm from India, curcumin is really good, but as a drug, when you are testing as a drug, you have to be extremely careful, and that is the case with another compound called berberine. Is an isoquinoline natural product, and it is also been tested and works for every disease. But it has a toxicity because of insolubility and induce mitochondrial fragmentation and so on and so forth. And we modified this particular molecule, and indeed, it protects the cells, under Alzheimer conditions and become multifunctional molecules. So that is about A beta and then other toxicity pathways around it. And we also working on tau. Well, we know that there is something called liquid-liquid phase separation. This is a natural physiological phenomena. So it happens in the cell to form a membraneless organelles. You can call it as a droplets, wherein by forming the droplets, it can increase the local concentration of any biomolecular biomolecule. For example, it can be protein. Here it is tau, and because of the increased concentration, perfect. So because of the increased concentration, it undergo aggregation to produce, to form a, protein aggregates. And here what we did was, kind of developing its own medicine, okay, using how these are the chemical grammar. What I show is that is a chemical grammar how to undergo aggregation. Can we use the same information in a, kind of a small molecule and prevent this, aggregation? So precisely that's what we did, particularly that particular compound what you see there on the side. So it forms a, droplets, and these droplets seeding into formation of aggregates, and now using a particular specific designed compound, we can inhibit the formation of tau aggregation and protect cells. Well, going through all of it, we learned a lot over the years, in terms of understanding individual pathways as well as a, kind of a holistically, and we come up with a molecule that is PGR63. This is a work of almost more than 10 years, actually 12 years of work, and indeed it become a very effective drug candidate. What you see here, that is a healthy cell. This is, we have a transgenic model which actually produce these protein aggregates in the brain, and then in the brain it mimics exactly like Alzheimer patient, both in terms of its pathological symptoms as well as cognitive symptoms. So what you see here, this is the wild type brain, and this is the AD brain. Particularly, I draw your attention, the red dots, what you see, those are the protein aggregates. Trust me, you take any patient, AD patient, and look into his brain, it looks exactly like this. See, so much of aggregates. And now we have this particular molecule TGR63. We treat these AD mice with TGR63, and there is a significant reduction in this amyloid burden to an extent of 75 to 85% in cortex and hippocampal region. Well, that is, this is definitely a good news. However, that is not enough. Just clearing the aggregates. In fact, some of the criticism with the antibodies is that it helps in clearing aggregates, but that doesn't translate into cognitive, improving cognitive functions. Of course, we went ahead and we did a behavioral studies, not just one. We did 3 or 4 different behavioral studies, and indeed it showed that it is translating the burden. The reduction in the amyloid burden, it is very nicely translating into cognitive functions. Possibly I'll just let me play a video. So for example, one test I will show you. Seeing is believing. This is called Morris water maze test. What we do is a big water top, and then you can hypothetically divide into 4 quadrants, and in one of the quadrant we place a platform, a small platform. The circle is actually drawn for our reference, and it is just underneath the surface, and we take the mice, both healthy mice and wild type mice, and we leave them in the water. Okay, we train them, and as they swim around, and then they have to rest, and then if the learning is good, if their learning ability is good, they will identify this platform. And later on we remove the platform and see whether this mice remembers the place where this platform was, meaning we are testing its memory. Let us see what happened. So we have 2 sets of mice here, wild type. Again here vehicle means is not treated, and TGR63 treated as well, to see whether effect of our molecules on the healthy mice. And in case of Alzheimer, again in one batch we are not treated, another batch is treated. Yeah. Oh. So this is the wild type, and this is Alzheimer type. So there is a platform. See, they are extremely good in learning. They are normal, healthy mice. They already found the platform, and these Alzheimer mice, still they going around. See, this mice comes close to that. Still, it has no clue that there is a platform. And the treated one, drug treated one, indeed finds the platform, meaning there is a improvement in cognitive functions. And but this guy is like a vagabond, going around, like keep on going. Okay, there is an anxiety as well. This is a quantification. Now we remove the platform and see what happens, and test whether they can remember. So there is no platform, but just see that how many times this mice comes to that location. See, these healthy mice, they come several times, and so is the AD treated mice. But this guy again, no clue. See, it comes, they remember, there is a memory, very good memory in case of healthy mice, and also so is in case of the mice, AD mice treated with the drug. So that clearly established that indeed this not only helps in clearing protein aggregates, but also improve cognitive function. In fact, it became a very big news around, and several pharmaceutical companies come forward for the further development. And we identified one company from US, and now it has been licensed to them, and it's very unique molecule from India, completely novel molecule, novel entity, and this will be going for clinical trial very soon. Now before, I don't know how much time I have? 5 minutes, perfect. So now I will change the gears. Well, that is good. We have reasonable, good success so far, but that is still we don't know. We have another 2-3 years to go to test this, to see what happens in terms after the clinical trials. But meantime, we started looking into many mechanistic aspects. So one such mechanistic aspect is peroptosis. I don't know, you all must have heard programmed cell death, like apoptosis and autophagy, and then comes necrosis, which is necrosis, not a programmed cell death, and the version of programmed cell death version of necrosis is necroptosis, and so on and so forth. And recently, it was discovered another cell death mechanism that is called peroptosis. Again, I will not go into the detail, it's just a snapshot. So the Lebel iron pool, the free iron, most of the metals which is there in our body, they are in the bound form, then they are not toxic. However, if this iron is in the unbound form, that becomes toxic. Particularly, that's what I am referring to labile iron pool. These undergo pendant type reaction and produce excessive reactive oxygen species that oxidizes lipids. It's actually cause lipid peroxidation, and this lipid peroxidation can induce biomolecule damage and also mitochondrial damage and many other downstream upstream effects leading to cell death, that is precisely peroptosis. However, cell do have a repair mechanism. There is an enzyme called, there are many enzymes, antioxidant enzyme, and one such enzyme is GPX4, glutathione peroxidase, which under these conditions, if there is such oxidation, these glutathione peroxidase reduces these peroxides into lipid alcohols, which are less toxic. Unfortunately, under Alzheimer conditions or a peroptosis conditions itself, there is a down regulation of GPX4 and then peroptosis takes over. So here for the first time from our lab, it is a very recent work, we established that in fact iron in its free form are bound to A beta and tau, two of the toxic proteins implicated in Alzheimer disease, indeed cause a peroptosis like conditions in Alzheimer disease. In fact, this is the first example, although there was a talk about it, this is the first example or the first time we directly link peroptosis and Alzheimer disease. And of course, we went ahead, this is all the studies, I will not go into the detail unless you are interested, and indeed we kind of selected some of the polyphenolic compound, and we to interfere with peroptosis and Alzheimer disease together, and particularly this molecule TA, indeed probably I'll skip this, all of this, indeed bind to GPX4 at there is a GPX4, this has a two binding site, one is inhibitor site, another one is activator site, and tannic acid binds to activator site and enhances its activity, and also we found that there is an enhancement in its levels, and in that way it actually modulates both peroptosis and Alzheimer's together. Just to kind of a nutshell, that's what peroptosis, we have identified the link, between nexus, pathological nexus between peroptosis and Alzheimer disease, and now we have also identified a molecule which can interfere with these pathways and inhibit both peroptosis and Alzheimer disease as well. This probably I'll skip. To summarize, of course, we are working on many of the several mechanistic pathways. In fact, we also identify new non-coding microRNAs, which possibly I can talk about on some other occasion, which is completely new and which is useful for both target, it's a mechanistically interesting, and it is also useful for diagnostic and therapeutic purposes. So to summarize, I hope particularly for the student audience, so I convinced you that Alzheimer is not a simple disease, rather a multifactorial disease, and there are multi-biomarkers and targets. In fact, yesterday, I don't know how many of you paid attention to Dr. Vengama. It was very nice. She has nicely put up, the multiple biomarkers involved in various diseases, including the Alzheimer disease. So there are multiple biomarkers and targets. We need to be mindful, and differential detection and validation of also, coming up with new biomarkers which can be added to NIA-AAA list. And multifunctional, a multi-pronged approach is definitely useful in tackling Alzheimer, complex disease like Alzheimer disease. And I did show you a drug candidate which is success story so far, and hopefully we are crossing fingers so that indeed it become a successful and become that pill everybody is looking forward, because it is a small molecule, very, very small molecules, it will be cheap, and it can address both developing and developed world both. And of course, we also established GPX4 peroptosis AD axis, which is one of its kind, mechanistic study. And of course, let me thank my students, and also collaborators and funding agencies. Thank you very much for your kind attention. I will be happy to discuss with you and take suggestions as well, from you later on. Thank you very much.
Long live India and France friendship.